Hello friends. In this lecture, I will talk about bioenergetics and oxidative phosphorylation. Here are some questions on these topics. Coupling of the reaction centers of oxidative phosphorylation is achieved by which one of the following? Making a complex of all four reaction centers, locating all four complexes in the inner membrane, ubiquinones and cytochrome C, pumping of protons. Then another question was asked. A practical class was going on where the students were demonstrating ATP synthesis in vitro using active mitochondria. Some students added one of the following to their tubes: dinitrophenol, an uncoupler, mild acidification of the medium, glutathione that permeabilizes both the membranes, an outer membrane permeable proton quencher compound, allyla, in which one of the above ATP synthesis will be detected. So you have to identify the correct option. So let's first discuss on the basics of the bioenergetics and the oxidative phosphorylation. Life obeys the laws of thermodynamics. Therefore, to maintain their structural complexity, organisms must obtain energy from their environment. The energy cells obtain from their environment is most often conserved in form of ATP. ATP is used to supply the energy needed for certain chemical reactions to proceed. Oxidation reduction reactions play a critical role in energy conservation. Many of these redox reactions occur in electron transport chains. Cellular work and energy transfer. Cells, they carry out three major types of work: chemical work, transport work, and mechanical work. Chemical work involves the synthesis of complex biological molecules from much simpler precursors, where energy is needed to increase the molecular complexity of a cell. Transport work requires energy to take up the nutrients, eliminate waste, and maintain ion balances. Energy input is needed in this case because molecules and ions often must be transported across the cell membranes against an electrochemical gradient. Mechanical work is another kind of work where energy is required for cell motility and the movement of structures within the cells, such as partitioning of the chromosomes during cell division. If we talk about energy, energy may be defined most simply as the capacity to do work. Organisms, they obtain the energy they need from an energy source present in their environment. They convert the energy from the source into a useful form. The most commonly used form of a cellular energy is the nucleoside triphosphate ATP. In addition, other nucleoside triphosphates and other high energy molecules are required for specific processes. To understand how energy is conserved in ATP and how ATP is used to do cellular work, some knowledge of the basic principles of thermodynamics is also required. The science of thermodynamics analyzes energy changes in a collection of matter, for example, a cell or a plant called a system. All other matter in the universe is called the surroundings. Thermodynamics focuses on the energy differences between the initial state and the final state of a system. It is not concerned at all with the rate of the process. For instance, if a pan of water is heated to boiling, only the condition of the water at the start and at the boiling is important in thermodynamics, not how fast it is heated. Laws of thermodynamics. First law of thermodynamics says that energy can neither be created nor destroyed. The total energy in the universe remains constant, although it can be redistributed as it is during the many energy exchanges that occur during chemical reactions. For example, heat is given off by exothermic reactions and heat is absorbed during endothermic reactions. However, the first law alone cannot explain why heat is released by one chemical reaction and absorbed by another. Explanations for this require the second law of thermodynamics and a condition of matter called entropy. Entropy is a measure of the randomness or disorder of a system. The greater the disorder of a system, the greater its entropy. The second law states that physical and chemical processes proceed in such a way that the randomness or disorder of the universe increases. However, even though the entropy of the universe increases, the entropy of any given system within the universe can increase, decrease, or remain unchanged. Units of energy. Two types of energy units are employed to specify the amount of energy used in or evolving from a particular process. A calorie Calorie is the amount of heat energy needed to raise 1 gram of water by 1 degree Celsius. For example, raising the temperature of water from 14.5 to 15.5 degrees Celsius. The amount of energy also may be expressed in terms of joules, the units of work capable of being done. 1 calorie of heat is equivalent to 4.1840 joules of work. 1000 calories or a kilocalorie is enough energy to boil 1.9 milliliters of water. A kilojoule is enough energy to boil about 0.44 ml of water or enable a person weighing 70 kg of the person to climb 35 steps. Free energy and chemical reactions. The first and second law of thermodynamics can be combined in a useful equation relating the changes in energy that can occur in chemical reactions and other processes. Delta G is equal to delta H minus T delta S, where delta G is the change in free energy, delta H is the change in enthalpy, T is the temperature in Kelvin, and delta S is the change in entropy occurring during the reaction. The change in enthalpy is the change in heat content. Cellular reactions occur under conditions of constant pressure and volume. Thus, the change in enthalpy is about the same as the change in total energy during the reaction. The free energy change is the amount of energy in a system or cell available to do useful work at constant temperature and pressure. Therefore, the change in entropy is a measure of the proportion of the total energy change that the system cannot use in performing work. Free energy and entropy changes do not depend on how the system gets from start to finish. A reaction will occur spontaneously if delta G is negative. That is, without any external cause, if the free energy of the system decreases during the reaction, then the reaction will occur spontaneously. It follows from the equation delta G is equal to delta H minus T delta S that a reaction with a large positive change in entropy will normally tend to have a negative delta G value and therefore occurs spontaneously. If delta G is positive, then the reaction is less favorable, and if delta G is 0, then the reaction is at equilibrium. Free energy change of the reaction, which is a reversible reaction, depends on the concentration of reactant and product. At constant temperature and pressure, the free energy change can be given as delta G is equal to delta G0, which is the standard free energy change, plus RT ln concentration of B by concentration of A, where delta G0 is the standard free energy change, R is the gas constant, T is the absolute temperature, and concentrations A and B are the square bracket A and square bracket B are the concentration of reactants and the products respectively. Standard free energy change can be calculated from the equilibrium constant of the reaction under standard conditions, that is at a solute concentration of 1.0 molar, at temperature of 25 degrees Celsius, and at 1 atmospheric pressure. Free energy change that corresponds to this standard state is known as standard free energy change, designated as delta G0. What is the relationship between standard free energy change and equilibrium constant? For a reversible reaction A to B at equilibrium, the equilibrium constant is given as the concentration of the product at equilibrium divided by concentration of reactant at equilibrium. So at equilibrium under standard conditions, the overall free energy change delta G is 0. Therefore, the standard free energy change is given as minus RT ln B upon A. So delta G0 is equal to minus RT ln K. From this equation, we can substitute this with KEQ, which is equilibrium constant. So standard free energy change will be equal to minus RT ln KEQ. So if we talk about the KEQ and delta G0, then if equilibrium constant value is greater than 1, then the reaction will proceed in a forward direction. That is, at the same time, under same conditions, delta G0, that is standard free energy change, will be negative, and the reaction will proceed in a forward direction. When equilibrium constant is equal to 1 or standard free energy change is equal to 0, the reaction is at equilibrium. If equilibrium constant is less than 1 or standard free energy change is positive, then the reaction proceeds in a reverse direction. So this table is very important to remember these things. Many times questions are asked, uh, you may be given that if the equilibrium constant is greater than 1, then whether the reaction will proceed in a forward direction or a reverse direction, and so on. So this table will help you to memorize the concept of equilibrium constant, standard free energy change, and the reaction, whether it will proceed in a forward direction or reverse direction. Role of ATP in metabolism. All biosynthetic and polymerization pathways and possibly some assembly reactions require the participation of ATP or other compounds that contain reactive phosphate groups. ATP is a derivative of AMP to which two additional phosphate groups are attached through an anhydride linkage. These anhydride bonds indicated by the symbol approximate sign. So this is the symbol indicating the high energy bond. These are high energy bonds and thus are particularly reactive. Hence, ATP is able to donate phosphate groups to a number of metabolic intermediates, thereby converting them to active forms. Here is the figure of ATP, where the adenine nucleotide is attached to the ribose sugar, and this ribose sugar is attached to the phosphate, and to this phosphate, two additional phosphates are attached, resulting in formation of ATP. So these bonds are the anhydride bonds linking the beta phosphate and gamma phosphate. The phosphate which is directly linked to the carbon of the ribose sugar is referred as alpha phosphate. So the bond between the alpha and beta phosphate and beta and gamma phosphate, these are high energy bonds. When these are cleaved, high energy are released. So here ATP, when it is hydrolyzed to ADP, the free energy change of this reaction is minus 7.3 kilocalories. In terms of joules, it is minus 30 kilojoules per mole. When ADP is cleaved to adenosine monophosphate, the standard free energy change is also a greater one, where it is minus 7.3 kilocalories or it is minus 30 kilojoules per mole. However, when AMP is cleaved to adenosine and phosphate, standard free energy change is less. It is about minus 3.4 kilocalories or it is equal to minus 14 kilojoules per mole. Biochemical mechanisms generating the ATP. These involves substrate level phosphorylation or the oxidative phosphorylation or photophosphorylation. Substrate level phosphorylation involves the transfer of phosphate group from high energy compound to ADP, resulting in formation of ATP. So this kind of ATP synthesis occurs when the organism is carrying out the fermentative type of metabolism. Generation of ATP by electron transport chain occurs during the oxidative phosphorylation, which occurs in case of respiration, that is aerobic respiration or anaerobic respiration, and electron transport chain also operates during the photophosphorylation process, that is during the photosynthesis. Here, in the electron transport chain, the electron passes through several electron carriers and finally is accepted by the terminal electron acceptor. So here you can see that a primary electron donor it serves as the energy source. When it is oxidized, the electrons from the electron donor are accepted first by the primary carrier, the first electron acceptor, which gets reduced. This transfers the electron to the another carrier and itself gets oxidized, whereas the second carrier gets reduced. The reduced second carrier then donates electron to the third carrier, which in turns get reduced and the second electron carrier gets oxidized. The reduced third carrier then transfers the electron to the terminal electron acceptor, and the terminal electron acceptor gets reduced and the third carrier gets reoxidized. So this is how the acceptance of electron and donation of the electron from the one carrier to the another carrier results in redox processes. So the electron transport chain involves several redox processes from primary electron donor to the terminal electron acceptor. So this is how the electron transport chain operates in the organisms, where the carriers are located in the membranes, in the cell membrane of the microbes, whereas in case of eukaryotes, the electron carriers, they are located in the inner membrane of the mitochondria. So electron carriers and hydrogen carriers, the orientation of the carriers in the membrane is such that they transport in the direction towards outside the cell, and orientation of the electron carriers is such that they transport towards inside the cell. So this is the inside cell and this is the outside the cell. So this is how the cell, the electron carriers and hydrogen carriers are located in the cell membrane, and this is how they will be oriented in the membrane. In case of the mitochondria, the electron carriers are located in the inner membrane, and this will be the matrix side of the mitochondria, whereas this will be the intermembranous space, that is the space between the inner membrane and the outer membrane. Proton motive force. The cell membrane is otherwise impermeable to protons. As a consequence, electron transport traps a portion of the chemical energy released by the net reaction of the chain. Oxidation of the primary electron donor by the terminal electron acceptor in the form of a gradient across the membrane of protons and electrical charge. Such a gradient, termed as a proton motive force, is a form of potential energy capable of doing work. It drives certain permease systems that concentrate externally supplied substrates within the cell. It provides the energy for flagellar mediated cell motility, and it drives the energy requiring synthesis of ATP from ADP. Synthesis of ATP by proton motive force. The synthesis of ATP at the expense of the proton motive force is catalyzed by a complex membrane-bound enzyme called as ATP phosphorylase, phosphohydrolase, or sometimes it is termed as ATPase or ATP synthase. ATPase is composed of two multi-component proteins, F0 and F1. In case of bacteria, these components are referred as BF0 and BF1 respectively. The alpha and beta subunits of the BF1, this is the BF1 and this is the BF0 that is shown embedded in the membrane, whereas BF1 is oriented on the inner side of the cell membrane. So in case of mitochondria, the F0 component is embedded in the inner membrane, whereas F1 is oriented towards the matrix side of the mitochondria. So the alpha and beta subunits of the BF1 are arranged alternatively to form a hollow hexagon, the central hole of which contains the gamma subunit associated with other subunits delta and epsilon. So these delta and epsilon subunits are there and the gamma subunits are there, and alpha beta units are arranged alternatively in a hexagonal manner. This forms a hollow structure through which the protons can be pumped inside the matrix of the mitochondria or inside the cytoplasm of the cell, microbial cell. Thus, BF1 probably has the subunit structure alpha 3, beta 3, gamma, delta, epsilon. The alpha and beta subunits form the catalytically active portion of the structure, the site where ATP is synthesized from ADP and inorganic phosphate. The gamma, delta, and epsilon subunits form a proton translocating stalk and gate that bring to the active site at the proper rate the protons that drive the reaction. Electron transport chain and oxidative phosphorylation. Energy released during oxidation of glucose to carbon dioxide is retained in the reduced coenzymes NADH and FADH2 generated during glycolysis and TCA cycle. the electrons are then transferred from NADH or FADH2 to molecular oxygen through a chain of carrier molecules with fixed orientation in the bacterial cell membrane. In eukaryotic cells, these series of electron carriers are present on the inner mitochondrial membrane. So this is the mitochondrial membrane that is shown, outer membrane and the inner membrane. And you can see the complex 1, then complex 3 and complex 4, they are embedded in the inner membrane of the mitochondrial membrane. Whereas this is the matrix, you can also see that ATP synthase is embedded in the membrane, inner membrane. This is the F0 component and this is the F1 component of the ATP synthase enzyme. The carriers in an electron transport chain participate in a series of reactions of increasing reduction potential values designated as E-0 values between that of primary electron donor and the terminal electron acceptor. So this is the redox tower that is shown, where the different redox couples are shown and their reduction potential values are mentioned on this scale. So here the electron can move from the redox couple, which is having a negative E-0 value, to a more positive E-0 value or more positive reduction potential value. So here you can see that the NADH can give electrons to the quinones. Okay, so this is the reduction. This is the ubiquinone that is shown over here, which has the reduction potential value of plus 0.11. So this is the more positive one compared to the NADH. Okay, so the electrons can move from NADH to the reductase complex and then to the ubiquinones and then to cytochromes and so on. And finally, the electron can be accepted by the molecular oxygen, which has a reduction potential value of about plus 0.8 volt. So remember this thing, that the electron donor has a more negative reduction potential value compared to the electron acceptor. So electron acceptor has to have a more positive reduction potential value than the electron donor. Then and then only the electron can be accepted spontaneously. Or if the electron donor is having a more positive value than the electron acceptor, then energy has to be input for traveling the or for transporting the electron in a reverse direction. So that will consume more energy. Bacterial electron transport chain. The generalized electron transport chain in bacteria is shown in this figure, where the electron donor can, from the electron donor, the electron can be given to the dehydrogenase complex, which in turn gives electrons to quinone, and the electron then gets transported to BC1 complex, cytochrome BC1, and then to the cytochrome C, then to the oxidase, and then to the terminal electron acceptor. So the electrons can be pumped at various locations from the electron donor depending on their reduction potential value. So one electron donor can give electron to quinone, and which in turn gives electron to the oxidase complex, okay, which is turn then gives electrons to the terminal electron acceptor. In case of mitochondria, the electrons from the NADH goes to the complex 1, and from the complex 1, the electrons are accepted by the quinones. The reduced quinones then give electrons to the complex 3, and from complex 3, the electrons go to the cytochrome C and then to the complex 4, which in turn gives electrons to the molecular oxygen, which gets reduced to water. If the electron donor is FADH2, then the electrons are received by complex 2. Through complex 2, the electrons are pumped to the quinones, and then the electrons are given to the complex 3, cytochrome C, and complex 4, and then terminal electron acceptor, that is molecular oxygen. So here, if we look at the complex one, it has the prosthetic group FMN, iron sulfur proteins. Then complex 2, which is called as succinate coenzyme Q reductase, it has FAD and FES. Coenzyme Q, reduced coenzyme Q and cytochrome C reductase is called as complex 3, which has heme B 566, heme B 562, heme C1 and iron sulfur proteins. Cytochrome C oxidase has complex 4, which is it is called as complex 4, which has a heme group and copper ions. Components of electron transport chain. Electron transport chain of aerobic chemoheterotrophs are the most thoroughly studied, and these are composed of 4 different classes of molecules: flavoproteins, which are called as hydrogen carriers; quinones, which are also called as hydrogen carriers; iron sulfur proteins, which are electron carriers; cytochromes, which are electron carriers. Hydrogen carriers, they carry electron and proton. The protons are pumped to the inner membranous space in case of mitochondria, whereas in case of uh, the bacteria, the protons are pumped into the periplasmic space, and this generates the proton motive force due to the accumulation of protons in the intermembranous space or in the periplasmic space. Whereas electron carriers, they simply transport electrons from the electron donor to the next electron acceptors or next electron carrier. Inhibitors of electron transport chain. Several site-specific inhibitors are identified. These compounds prevent the passage of electrons by binding to a component of the chain, blocking the oxidation reduction reaction. Example is rotenone, which is a plant product which inhibits the transfer of electrons to the NADH coenzyme Q reductase complex. It is used as a fish poison and as insecticide. So rotenone will block this at this point. It will block electrons movement from NADH to ubiquinones. Cyanide, azide, or carbon monoxide, they bind with the cytochrome oxidase complex and inhibit the terminal transfer of electrons to molecular oxygen. So the cyanide, azide react with the oxidized form of the cytochrome, and carbon monoxide, it reacts with the reduced form, thereby blocking the transport of electron from the oxidase complex to molecular oxygen. Plants also have alternative mechanisms for oxidizing NADH, and electrons from NADH directly transfer to ubiquinone and from ubiquinone directly to molecular oxygen, thereby bypassing the complexes 3 and 4, and hence NADH oxidation by this pathway is cyanide resistant. Let us have a look at the questions now. Coupling of the reaction centers of oxidative phosphorylation is achieved by which one of the following? Making a complex of all 4 reaction centers, locating all 4 complexes in the inner membrane, ubiquinones and cytochrome C, pumping of protons. So here you can see that the coupling of the reaction centers of the oxidative phosphorylation can be achieved by ubiquinones and cytochrome C, because these are the couplers of the complexes. These are mobile elements or mobile carriers, so they can get mobilized from one place to another place within the cytoplasmic membrane or in the inner membrane of the mitochondria, and so these are the correct options: ubiquinone and cytochrome C. Next question is, a practical class was going on where the students were demonstrating ATP synthesis in vitro using active mitochondria. Some students added one of the following to their tubes: dinitrophenol, an uncoupler, mild acidification of the medium; glutathione, that permeabilizes both the membranes; and outer membrane permeable to proton quencher compound, Alila. So here dinitrophenol is an uncoupler, and so it will block the electron movement to the ATP synthesis. So the ATP synthesis will be blocked in this case. Mild acidification of the medium can help in the ATP synthesis. Glutathione, which permeabilizes both the membranes, will cause the gradient of the proton get disturbed. So the proton motive force will not be generated in this case. And an outer membrane permeable H plus quencher compound, if it is added, then proton motive force will get diminished. And so the correct option is B, where mild acidification of the medium will cause the ATP synthesis to occur. So the correct option is B, that is 2. So that was all about the bioenergetics and oxidative phosphorylation. Thank you and happy learning.